Good morning, everyone. Happy Saturday. Everybody hearing and seeing me okay? If you can hear me, give me a thumbs up in the chat or something here. Make sure we're actually broadcasting. All right. Starting to think I was talking to nobody here. Okay, I want to give you a little bit of background about myself here. I've been a rocketry hobbyist for about 45 years, but my educational background is in biology. I study the limits of life and look at extreme environments as analogs for targets elsewhere in the solar system and beyond, looking for the possibility of extraterrestrial life. And I've done this in laboratory settings, in field settings, most recently caves. And I was getting to the point where my body was just starting to break down. I couldn't safely do a lot of cave exploration anymore. And then COVID came along and kind of cinched it. And so I've been retooling my research now into looking at airborne microbes, and some interesting possibilities have come up in the last few years showing that microorganisms are not just being carried by the wind, but can actually metabolize and reproduce and grow while in the air, especially if they can latch on to clouds or they can have little water droplets clinging to them. Okay, so LADCAP is a dust collector. And what we collectively call dust is actually lots of microscopic and very small macroscopic particles. Of course, as we see here, bacteria and other microbes, these are what I'm particularly interested in, as well as their spores. And the difference here is a spore is a resting stage of any type of microbe, whether it be a bacterium or a fungus, things like this. Many different microbes have resting stages that we collectively call spores. And these are especially important because they're very resistant to environmental detriments. And these are often what get transported in the form of dust. Okay. Pollen makes up a large proportion of dust, especially seasonally in various areas as different plants flower and pollinate. And then cellular debris. So much of what we call house dust, about 50% of that is just the remnants of your own skin cells. A lot of small invertebrates live on those skin cells, so what we call dust mites. Some of you may have heard of tardigrades or water bears. These are another very, very small organism, but they're a true animal. And then we get a lot of inorganic stuff as well, or non-living. I won't say necessarily inorganic, but pollutants. Whenever you see a big rig truck take off and that big old black smoke comes out, it's mostly carbon. Volcanic ash. This becomes of particular importance in and around an active volcanic area. Often airplanes will have to be diverted around dust clouds, ash clouds, things like that. I was actually living in Washington State when Mount St. Helens blew back in 1980. That was an impressive amount of dust in the atmosphere. Okay, small mineral grains. This actually relates to volcanic ash. And it actually turns out micrometeorites. Every day, Earth's atmosphere receives on the order of about 40 to 100 tons of space debris in it. And some of this ends up in the dust in our atmosphere. So at this very moment, you could be breathing in little bits of space dust. So these are some examples in the extreme of atmospheric dust. Here on the left, this is the remnants of a dust storm coming off of Africa. And so this contains a lot of very fine mineral dust. In some cases, pollen will actually do kind of the same thing. If you get an area that has a sudden massive pollen release, you can see these types of clouds, not usually this big. If you go into, say, a heavily forested area, pine forest, things like this in about June, and a little wind whips up, sometimes you'll see this big yellow cloud come off the trees. This is an example of an active volcano. This one's in Alaska. And again, we get a lot of ashy material here. The really heavy ash falls out usually within a few tens of kilometers, but the ultra-fine ash can actually get up into the upper atmosphere, and that can circulate worldwide. If we apply this to other planets, we do know that Mars periodically has global dust storms, and in fact, this is probably what led earlier astronomers to believe that there were seasons with plants and maybe even crops on Mars. What they're actually seeing is the obscuration and then reemergence of dark and light patches on Mars. Now, quite some time ago, this was my very first scientific paper, and this was written in between the Viking program, which was launched and landed in the mid-1970s. It ended at about 1982. And then we had this big hiatus in Mars exploration up until the late 1990s, and we didn't land on Mars again until 1997. But in this, we were speculating that if there actually were still extant life on Mars, that dust could be used to carry nutrients to these communities and also to allow dispersal of spores or microbes and such. Unfortunately, once we got back to exploring Mars, we found that the conditions were even worse than we previously thought. And this is unlikely today, unless there are very, very hidden areas that are well protected. But it still emphasizes the importance of wind as a carrier of cells, of spores, of things like this, especially in areas that don't have water flow. If we go up on the space station, dust is a problem for several reasons. This particular experiment called MAIDEN is actually looking at the repulsive effect of certain fabrics and treatments. And the idea here is to prevent microbes from building up in the nooks and crannies, not only of the instrumentation, but of the astronauts' clothing. Since the astronauts are breathing continually recycled air, continually filtered water, at least, if not completely recycled right now, they want to have ways of keeping the station as clean as possible so that the filter systems that are in place can work to maximum efficiency. Now, I'm not the first to come up with methods of using rockets to sample the air. In 1999, these two came out. The one on the left here is part of a short book called Second Stage Advanced Model Rocketry, which is actually referenced by this paper, which is available on the NAR site. Both of these use passive methods. So essentially they've got a sticky substance. In the case of this one, they use an agar-coated slide. In this, they use a kind of a sticky filter pad. And in both cases, it's actually the movement of the rocket through the atmosphere that is capturing the microbes. So in both cases, basically as the rocket moves through the atmosphere, any microbes or any other particles for that matter are slamming against the collection medium. And this works great. And in fact, in some ways, it works better than my system that we'll see because these get a lot more airflow. What they don't do is give you a calibrated airflow. So if I want to know how many microbes are in, 10 liters of atmosphere, my system can do that by simply turning on and off at appropriate points. Both of these, basically, they're open during launch and then they close during recovery. And so you're getting sampling through a column of air. And again, there's nothing wrong with that, but it does provide some limitations on experimental design. So there's no, what I want to say is there's no one way to do this that's going to work for every situation. Okay, so this is the basic system, and there's really not much to it. So this is something that anybody can build, anybody can use. What you see here in the photo is $30 or less to build. And it's very small, so you can fit this into a 4-centimeter diameter airframe. Depending on the actual motor you use and the pore size of the filter, I get about 2.5 to just under 3 liters per minute throughput. Okay, so that's simply how much air you can suck through this thing here. This little part is a membrane filter holder. This particular style, it uses what's called a lure lock system. This is the same thing that we use in medical supplies for attaching syringes and IVs and everything. It's a standardized miniature plumbing system, basically. Okay, the whole thing's powered by a lithium-ion 9-volt battery. You can use alkaline if you have to. It just weighs a bit more. But I actually did this purposely so that batteries are available easily if you have a problem with them. I know there are a variety of lithium-ion battery packs that are custom made. They're flatter, they're wider, whatever have you. But at the end of the day, if you lose one or you forgot to pack it, it's harder to find those than it is a standard 9-volt. Originally, I had envisioned this using a micro switch that would activate this pump either at launch or at apogee. And so in its most basic form, it does act a lot like the previous two I showed you there, where you either turn it on right before launch and you fire it up, and then when the ejection charge goes off, the switch would release, and then that would turn the pump off. Or alternatively, you could do it in reverse. The switch can be put into a normally open or normally closed configuration so that you could launch it up in the normally open, or excuse me, in the normally closed. When it's being launched, the switch is depressed to keep it open, and then at apogee, it gets released. The little bar here gets released from the payload, and that turns it on, and it collects all the way down. I built this mainly for testing. It's never actually been flown in this configuration because what I really wanted to do was have the ability to turn on at a particular altitude and then turn off. So I'm sampling a particular layer of air. Okay, and this is in general what filter type systems look like. So this is actually a larger version, but works on the same principle. So the idea is that as air is pulled through the filter, the microbes can't go through the filter. The filter size is smaller than the cell diameter. And then that is placed onto some sort of nutrient medium, nutrient agar or whatever. We'll talk more about the biology a little bit later on. But then each one of those cells grows into a colony that you can actually look at here, and from there you can isolate those colonies, do biochemical tests, DNA identification. You can get really fancy with this or really basic. If you're fortunate enough to have an electron microscope, you can actually take a portion of that filter and directly view what's on it. And so the little holes you're seeing here, these are the actual pores in the filter membrane. These are bacteria here. I don't have this kind of setup. Most people don't. But if you have like a big research university nearby that would be willing to let you have some scope time, this is definitely a possibility. Now we'll see later on for bigger things like pollen and dust mites and things like that, you can do this with a relatively inexpensive optical microscope. Okay, so we wanted to first of all just make sure the filter system worked. So we did a series of ground tests where we simply turned the pump on for a set period of time and looked at how many bacteria we captured. And what we came up with was we get about 1 colony forming cell per liter of air that we filter through. So for example, in this particular case here, this ran for, I think, about a minute and a half on this one. And we ended up with 2 colonies. So presumably each colony here grew from a single cell. Now, something to keep in mind, there is no single type of growth medium that will grow every type of cell. And so we actually have to decide on what we're going to grow this on and look for that particular target type of organism. So there's no universal growth medium, unfortunately. And I'll talk more about that later on as well. Okay, so let's talk about the actual lifting platforms here. When I was first designing this, I had 3 basic types of platforms in mind. I've got a colleague that does a lot of what are called balloon sats. And so this is where you take a weather balloon. It usually has some sort of telemetry package here beneath its parachute. This is usually at minimum a GPS locator. And then you can have experiments that daisy chain beyond this. And so LADCAP could be included as one of these. Rockets, of course. I designed the original LADCAP to fly on mid-power rockets. Other versions can fly on higher power rockets that have more capability. And then higher end drones, not the multi-$1,000 ones, but the $1,000 to $2,000 ones have enough lifting capability to use this as well. And all of these have their own benefits and drawbacks to them. So first of all, what altitude do we want? And this is kind of fuzzy because of some uncertainties, but also, especially in the case of rockets, we've got a wide range that we can fly to. Balloon saps typically have a ceiling of about 20 to 30 kilometers. This is when the pressure outside is so low that the balloon finally pops. And you don't have a lot of control over balloons. Once you put them up, unless you're using a tether on them, and that's only good for maybe a kilometer or 2 at best, you have no idea where they're going to go, which is why they always had GPS units on these things. And they still lose quite a few of them. Okay. But you can have a flight time and a sampling time of hours. In some cases, maybe half a day or more. But the initial outlay for this, you're looking at about $1,000 for the basic telemetry, for your first balloon and parachutes, all this kind of thing. So that mostly you're looking at here can't talk now. The instrumentation and the stuff to recover it all. The launch cost is surprisingly high. This actually caught me a bit off guard when I found out about it. And this is mainly because helium has skyrocketed. Strangely enough, helium is the second most abundant element in the universe, but on Earth it's pretty rare. And it's actually mined. It sounds really kind of strange, but helium on Earth is mainly a product of radioactive decay. it gets caught in pockets in the Earth's crust, and we actually mine that. And in the U.S., almost all of our helium comes from a mine in Texas. Okay, so every one of your launch, the balloon itself runs about $50 to $100, and then you get about $500 worth of helium that has to go into that. If we look at the rocket, here we've got a wide launch range. So depending on the size of the engine installed, the mass of the rocket, things like this, we can actually vary this a lot. And the 5 kilometer upper limit here, that's just being really conservative. I know there have been experimental rockets that basically reach space. Now, normally rockets don't drift as far as balloons do, although if you launch one up to 5 kilometers and you have a parachute up there with no falling period on a drogue, it can still drift quite a bit. But here we're looking at a sampling time usually between 1 minute and 15 minutes. We could go higher, and we'll look at some of the options for that a little bit later in the talk here. But the initial outlay here is about $100 for a mid-power rocket that's set up with, say, reloadable engine casing and payload capability for the device itself. And then the launch cost is basically the cost of your motor. So, if I'm launching this on an F, it might be $15, $20. If I'm launching it on a J, it might be up around 100 or more. Okay. And I'm actually going to talk a little bit about the conversion of a Hi-Tech into a carrier for this, but also give us a look at a smaller rocket that was used for prototyping. And then finally, the drones, they actually do have a very limited altitude, mainly because of one, FAA regulations, but also the remote control range of the transmitter. Horizontal range is about the same. It's line of sight, so usually you can go a little bit further vertically than you can horizontally. But the nice thing about the drones is you can control this throughout the entire flight, assuming you don't lose signal somewhere. And so you know exactly how far, you can take it out, bring it back to where you are. You don't have to chase it down. And you can actually do prolonged sampling at a given altitude with a drone that you can't do with a rocket. But of the three, this has got the highest initial cost. To get a drone that's actually going to be powerful enough to do this sampling for a long period of time, you're going to have some outlay. And here I give an example of the DJI Phantom Pro, which is the photo I showed earlier. Okay, so this is the first test rocket we used. This turns out it looks a lot like a miniature version of the Hi-Tech. That was completely coincidental. I built this before I even knew the Hi-Tech existed. But this, the whole thing is less than a meter long. It uses an Inticore AIM flight computer, not for deployment, but to control the payload. And as it is here, it uses a single 25 millimeter diameter filter. Pretty light, and so this can be flown without a waiver as long as you're flying G or below. And so we can run this with an engine as small as an E23. Can run it smaller, but you don't get enough sampling time. So this gives us just enough sampling time where basically we're sampling almost all the way up and all the way down. Initially, this was built only to use mid-power rockets. Now we'll see a variant of this that does away with that. But with a G64, this gives us a maximum altitude of just over 1 kilometer and a really nice flight time. Okay, so if we take a look at this in schematic form here, it's a pretty basic rocket, but we've got an avionics bay that's built into the payload coupler here. And then the payload bay is where we've got the vacuum pump, the filters. The actual battery is housed in here with the computer. And so packed for launch, this is how the payload bay looks. Now, one of the problems with this, just to try to packing it, the vacuum pump ports, the outlet or the inlet port for the vacuum is on the side, the outlet port is in the middle. And I really wish someone would come up with one that does the opposite. But basically all of these vacuum pumps are built the same way. But if it was in this, if the vacuum inlet was actually in the center of the pump, I could mount the filter directly onto the pump. I can't do that here. Okay, if we explode this outward, kind of look at everything here. So here you can see the pump filter. There's a little tube here that hooks on to a little port on the side. There's another port on the opposite side. This is just the air exhaust for the pump. So the air is simply exhausting out of here. It doesn't actually need a tube or anything. It's just as it pops out or pushes air out of here, it just vents out of the payload bay. And this is just so that we don't have air seeping into the cracks and such and into the avionics bay where it could cause problems in altitude recognition. So if we start pumping air into this thing, it'll think it's at a lower altitude than it is. I used this particular flight computer because most dual deployment altimeters have a set duration on their output. So because really they're just made for deploying, and all I have to do is ignite a charge. And so their channels are only open for a second or two, sometimes even less. The Enticore, you can program the duration of each of the two channels. They have a maximum duration of 25.5 seconds. And so if you run these in parallel, which I did, you can get about a 50-second sampling time here. I'm still working on a way to get more out of it than that, but right now it works well for testing. And so it had its maiden flight earlier this fall. Actually came out really close to its predicted flight. And I've got a short clip of that. I've got the sound turned off on this. Back into focus there. Okay, so the sampler turned on about 2 seconds after launch and then remained on until just before touchdown. And so all during its descent here, it is on. It's sampling here. The spin was a little bit annoying, but actually was probably beneficial to the actual sampling mechanism. And unfortunately, then it landed in an oak tree. Okay, but for the most part, it worked as planned. So we didn't have any problems with the rocket itself. The flight computer worked perfectly. The sampler turned on when it was supposed to, turned off when it was supposed to. But we did lose a fin on it. And once I got it back to the lab, I found, much to my chagrin, that there was a piece of backing paper that had gotten left on the filter membrane. And so we didn't get any sample. We did go ahead and culture the membrane anyway and found nothing on it, which was good, which means we weren't having any problem with contamination. Okay, but it was a bit broken, and so I was actually able to remove this. I thought I was going to have to toss the whole thing as it's a through-the-wall fin mount, but we were able to scrape all the pieces out of it and rebuild that. But it showed that maybe we needed a little bit heavier material. So we did rebuild the original one here, but then replace the payload bay. This is being used now for dual deployment experiments on a small scale, and I'm not going to worry about talking about it the rest of the time here. But the yellow one here, we rebuilt the fin can, replaced the balsa with plywood, and then modified the engine mount so that it can accept up to H-size engines. A few other things, replaced launch lugs with rail buttons, and now this has been completely redone. It's ready to fly. We've just been waiting for good weather now. Every time I try and go out and fly, it's either windy or raining. Okay, dimensions are essentially the same because we use the same sized airframe tube on that, just changing the fins there. So the payload's the same. We got a little bit more mass here, mainly because of the plywood fins. But it didn't cost us too much. So projected flights are still pretty good here. So we lost a couple tens of meters on the lowest size. And then now we can run up to an H-size motor in this and get almost 2 kilometers. Okay, so here again, a plan view of this. As I said, mainly the improvements here are we just made the engine mount capable of holding a high power engine, more robust fins there, but everything else still works the same. Okay. Now, when we first came up with this idea, we realized that mid-power will be good. It'll get us, as I said, up to about 2 kilometers. But to really get up the atmosphere, we're going to need something higher powered. And so I had actually, about the time that we were kind of looking at different high power kits, I was working on my own level 1 certification. I was building this thing mainly for the experience, not necessarily for as a scientific carrier yet. But as we got to looking at it, one of the things we found is that we could bump up the filter size to 47 millimeters, which is the next standard size of these things. And these fit almost perfectly within the payload bay. And the Hi-Tech actually comes with a good sized payload bay. And so now we could use, still use mid-power engines if we needed to, if we didn't have a high power field available. But this will accept up to a J-size engine, which gets us up to 3 kilometers almost. Okay, and the big thing here is we can do almost 3 kilometers with 4 times the sampling capability. And so in this case, we can use a little more complicated flight computer that will allow us to control multiple pumps. And then we can have filters up in here in a variety of configurations. So this is one. And here we've replaced the standard payload bay with a clear one. These are the 47 millimeter holders. And one possibility here is you can actually run 4 filters in series, and these can be set up so that you have a very coarse filter here that only captures really big stuff. And then as you go through each one, it becomes finer and finer. And this allows us to do differential sampling all at once. And that's one possibility. Inside the avionics bay here, we can mount 2 of the same type of vacuum pumps that we had in the original prototype. And so you can either use both of these in parallel to do this kind of sequential sampling, or we can run some tubing up here and run individual filters off of each vacuum pump. With this setup, we'd be limited to 2 filters in here, but we'd still have lots of room. Or you can kind of split it and say we want to do a 2 filter differential type of setup. So a coarse and a fine filter on one pump and another coarse and a fine filter on another pump. Lots of variations here. The other thing that will at least fit but requires a bit more complex tubing manifold is we can stick 8 of the smaller 25 millimeter filter holders in here. So this has the potential to doing much more complex filtration and sampling. Okay, so this is just a quick comparison. I'm not going to read all this to you. Some things to look at here. The altitude gains. Basically, we get 1 more kilometer using the Hi-Tech. But again, keep in mind that this is 1 more kilometer, but with a much larger and much more complex payload. If we were just flying the original LADCAP single filter type, we could easily break 3 kilometers here. Okay. Acceleration here we need to be a little bit wary of, simply because the tubing that connects some of this stuff, if you get your acceleration up too high, it'll collapse that. Okay. We also try and watch that we keep velocity down. We're not trying to break speed records here. What we prefer are nice, long, slow-burning flights that give us a lot of altitude, but not a lot of acceleration and velocity. Neither one of these rockets is really set up to do transonic flights. Okay, but both of these in their highest motor configuration give us nice long sampling times, which means we can either go up, sample for a long time on the way down, or even go up, do some sampling at one altitude, change to the other filter set, do some sampling at that altitude, and maybe even more complex setups where we can do, maybe every 500 meters we turn on a different sampling set, and we can actually get layers of atmosphere that way. Okay, the cost of prototyping these things, you can see the original G-Lifter and G-Lifter Mark II weren't too bad. Most of that cost is actually in the flight computer and the motor casing. In the Hi-Tech, we are planning on using a much more complex flight computer that has more channels, so we can turn on and off more of our pumps, and it also has an integrated GPS system in it. It's the Enticore Extra. Okay, now for future work, and this is still very much a work in progress. One is a more complex recovery system. And so many of you, I'm sure, are familiar with dual deployment, and this is what most flight computers are set up to do, where we want to have a nice high flight, and then we use a drogue chute at apogee, let the rocket fall quickly for a long time, and then have a main chute pop closer to the ground so you don't have to go chasing after it. But this is actually the opposite of what we want to do if we're atmospheric sampling. So that what we really need is a very large, in fact, an oversized parachute to deploy at apogee so that we have a longer time for sampling. And then after sampling is done, we want to somehow cut or reef that large parachute so that it acts more like a streamer, allowing the rocket to now descend very quickly now that we're done with the experimental part of it. And then we want another parachute that's a normal recovery size to deploy at about 500 meters or so, so that we can safely recover the entire package without having to have chased it down for, in some cases, several kilometers. Okay, and unfortunately, most rocket electronics out there that are set up for dual deployment simply won't work in this manner. Okay, another thing we want to look at is manifold sampling. So right now in the Hi-Tech model, we can have 2 different sample sets acquired, but we do that by turning on one pump and then turning on another pump. And that's, again, that's mostly due to a limitation in the flight computer. What would be more ideal, especially if we want to be able to do multi-layer sampling, is have a single vacuum pump here, multiple filters, and then have each one of those filter connect to a manifold with a small solenoid valve, so that the pump comes on and then we open one valve and close it, and open another valve and close it, and open another valve. And so each one of those valves could be stepped to sample through a filter at a different atmospheric level. And of course, with any rocket, we want more altitude. As we saw, the gain with the high-tech was not as much as you might think, but because of the increased mass of the more complex form of LADCAP, that's where a lot of that altitude went, is into mass lifting. Now, I do want to get back to the biology here. And as I said, this is actually a really good opportunity to do citizen science. You don't need to be a professional scientist to do this, although I will put in a few cautions here. Whether it be growing microbes or looking at pollen or dust grains or whatever, most of this stuff is relatively easy to do and not very expensive. A lot of it will require a microscope, but the type that you'd find in, say, a high school is more than adequate for that. So the first thing we want to do is look at what type of filter do we need. Now, for sampling air, the pore size here is much more important than the filter material. These different filter materials here are mainly for different types of solvents if you're doing this with a liquid. And so we broadly put them into what are called hydrophilic or water-loving filters and hydrophobic. And these are set up so that the hydrophilic ones, if you were filtering water or aqueous solutions, these are what you use. And then if you're doing filtering of really nasty solvents, acetone or gasoline, things like this, you would use these things. Okay. For sampling air, the mixed cellulose esters are easily used. They're very appropriate, and they're also the cheapest. When you get into these ones down in here, these can get really expensive. Nitrocellulose is similar, except this is basically flash paper or gun cotton. And so that you do have the possibility that you could have an accident with your rocket and burn up your filter at the same time. So generally, these are easily available. You can buy them on Amazon, and they're also the least expensive and also the safest. Okay, but all of these come in a lot of different pore sizes, and standard ones, standard measured in microns. And so if you want to look at bacteria or cyanobacteria, which are photosynthetic bacteria, you need the smallest pore size, because these are cells that are on the range of about half a micron to a micron in diameter. If you want to look at fungal spores or yeasts that might be in the atmosphere, here you can use a larger pore size. Now, the other advantage here to using larger pore sizes is you get higher throughput, so you can filter more air in a given period of time. And that's something you actually need to calibrate LADCAP to do. So you have to have a flow meter attached to it to calibrate it. Now, if you don't care how much you went through, if you're just going for something qualitative, you don't need to worry about that part. Okay. As we go up, if you just want to know kind of a general dust in the air, then you're looking at about a 1 to 10 microns. Pollen has a really wide range, so some pollen grains are enormous, microscopically speaking. Some can be really tiny. So here it kind of depends on the type of pollen you're looking to capture. Protozoa and algae, these are bigger cells, so about a 10 to 50 micron. And then if you're looking for actual invertebrate animals that are in the atmosphere, and they're there, dust mites, tardigrades, things like that, then you're looking at a much bigger pore size here. Okay, if you're going to do something with microbes, usually you're going to need to grow them because it's really difficult to just put a membrane filter under a microscope and look at bacteria, mainly because most bacteria are transparent. They don't have a lot of color, and they're very, very tiny. And so if you're not used to looking for them on membranes, which most people don't, it's really hard to find them. Okay, but various types of agar, and agar is just a gelling agent. Some people actually use it for gluten-free foods because without gluten, your breads don't hold up as well. It's also used sometimes in jam making, but it's just a gelling agent. Gelatin used to be used way back in the 19th century. Unfortunately, there are too many microbes that will eat the gelatin part, the protein in there, and then it all turns to goo. Okay, so if you're just looking for something to culture kind of general purpose human associated bacteria, then you can use things like nutrient agar, tryptic soy agar, which is basically vegan nutrient agar, one called R2A agar. These are all just general purpose media for general purpose microbes, if you like. If you're specifically looking for human pathogens, and here, if you're an amateur scientist, don't do this. And I'll get back to this again in a moment. But this is something that requires some training and precautions, and in some cases, some legal permissions to do. But now you become more specialized. There are things called blood augers, brain heart infusion auger, and it's just what it sounds like. These cost a lot more as well, and they're not always available to the general public. If you wanted to specifically grow fungi, the two most common here are called Sabra dextrose agar or rose bengal agar. This really pink one in the display here is rose bengal. If you want to look for photosynthetic organisms, there's one called BG11 agar. The BG is for blue-green, which is what cyanobacteria used to be called. And then if you want to get really fancy and look for what are called lithotrophic bacteria, and these last two are what I actually work on most of the time. These are organisms that eat rocks to gain energy, and are also related to what we find around deep ocean hydrothermal vents. And so both of these have interest in me from an astrobiological point of view. All right, then you've got to have something to grow them in. And if you're just doing general purpose culturing, the best thing to grow them in is a shoebox on a countertop. Okay, you just keep them in the dark, you grow them at room temperature. And the reason I say that is you're less likely to grow human pathogens that way. Or, you know, if you need specific things, so like when I'm growing algae and cyanobacteria, I use a lighted growth chamber, kind of like this one. But you can also do it just in a lit area where you've got indirect sunlight coming in, and you can just leave that Petri plate out there on a countertop or whatever, where it gets indirect light. You don't want really harsh light on it. And again, at room temperature, that's what most of these things grow at anyway. Okay. Now again, unless you have the education and proper facilities, try to avoid growing human pathogens. And since 9/11, you know, there might even be agencies out there that they start seeing you acquiring things for human pathogens that might set off a red flag, think you're a bioterrorist. Okay, as I said, for safety reasons, it's best to incubate these guys at room temperature. Okay, and here are a few more of our ground tests. So all these were grown on nutrient agar, just general purpose. And so sometimes you do get fungi. This is a fungal colony here. There's another one here, bacterial. This one is almost all fungus. It's hard to see it against the white, but there's a fungal colony there. There's one there. There's one there. There's a bacterial colony here, but it's got a fungal colony growing over it. Kind of the same thing here. Fungus, big bacterial colony there, and colony there. Okay, so we do know the system works. Now it's just a matter of getting more testing in flight done. Okay. Usually once you've got colonies on membrane, then you want to transfer those to new media to allow you to work with those, manipulate them, eventually identify them. And so these are just some various cultures here I chose mainly because they're easy to see. But these were all grown at room temperature or below. These guys up here are all grown at room temperature. These ones here are grown at a little bit lower. They're grown at about 13 degrees C, but they'll grow at room temperature as well. Now, once you've got these, if you want to actually see the bacteria themselves, you usually need to stain these. And again, this bacterial staining kits are available from educational supply houses, things like this. It's actually a fairly easy skill to acquire. For pollen, you can usually directly visualize these on the membrane. So you can actually just take your membrane out, stick it on a slide, put it under a microscope, and there are lots and lots of different shapes of pollen grains. And there are resources out there that actually allow you to identify the plant that the pollen came from based on what its pollen looks like. All right, some bigger guys, these are dust mites. These are crawling all over you right now. So if you get that little shiver, that's them. Again, bigger pore sizes here. This way you don't have to worry about trapping all the little microbes and such if you're just interested in the organisms themselves. And these also can be directly imaged on the membrane underneath an optical microscope. All right. As we wrap this up, I'd like to thank the Arkansas Space Grant Consortium that has continuously funded my work over the past 20 years or so. Very appreciative of the St. Louis Rocketry Association. They were the nearest club to me where I could get my level 1 certification done. And with that, I would love to take questions here in the Q&A. Looks like we've got a few. All right, excellent use of metric units, micrometers versus microns. Yeah, I know. But when you actually go to find these filters, they're usually found in microns. But I get you. All right. Okay, what are the best practices for keeping the filter clean pre and post flight? That I didn't go into. You need to have some way to sterilize these first of all. Okay, so what I do is I'll open the membrane holders screw together, so you just open those up. You put your filter membrane in, close them down, and then I wrap them in aluminum foil and put those into an autoclave. Now, if you don't have an autoclave, you can use a pressure cooker. Most pressure cookers will have a little rack that will raise them up off the water. You don't want these in the water, you just want them in the steam. And then once you take them out of the pressure cooker, just leave them out, say overnight, to allow any water that might have got in there to dry out. And then once you get out to the field, I usually load these. If my launch field's a short ways away, I'll just load the whole payload bay together at that time, put the filter holder in there and have it ready to go. Once it's loaded inside, as long as the pump is not operating, there's no flow between the ports, really. And so you don't have to worry about the casual microbe straying in there. It's not going to get in. Where do I launch at? Currently, I launch either on the Lyon College campus where I work, or I've got a friend who's got a small farm. I launch out there. I'm actually looking for better launch sites because these I can launch the E flights on, but the larger mid-power and up into the high-power flights, right now the only areas I know of that are launching is St. Louis and Memphis. Those are the two closest to me. When I was up at St. Louis doing my high-power certification, I was going to launch there, but it was so windy I was afraid of losing the payload. Missed that. Clicked the wrong one. They're about boat tails, but yeah, I've thought about boat tails on there, but right now I'm more interested in trying to get the whole payload package working. Let's see here. Can Inticore be programmed to turn on an apogee? Yes, it can actually be turned on based on timing, based on altitude. Can't remember. It's actually really versatile. The only problem with it out of the package is that the maximum duration of each channel is 25.5 seconds. Now, supposedly you can reprogram that to do longer, but I don't know how. Now, the Enticore Extra, which I'm planning on putting into kind of the next iteration of this, has no limits on the channel program. So, and it also has a GPS built into it. So it costs a lot more, but it also does a lot more. Okay, so let's see, probably similar question to Thomas's. Wouldn't you want, for some science purposes, to collect particles at specific altitudes? Yes, that's actually the entire reason that I'm doing this way, rather than the two previous designs that I showed there, which are basically, you know, all the way up. This one, you can program that Enticore to turn on at any altitude and then turn off again. Okay, for the high school level, how would you state the overall objective? In some ways, this is, if you want to do it from an engineering perspective, this is exactly how real space science missions work, is that you have a payload that you need to get to somewhere, whether it be Earth orbit, the atmosphere, Mars, wherever. And then you need to find what type of booster do you need on that, what type of performance is it going to do, and then is it going to match what you need to get out of your payload. And so if you have a specific experiment in mind, you could pick any of these that I've talked about. You can come up with your own. You know, so you might want to know how much pollen is in your air at, say, 200 meters, you know. So you could set this up to do that, take your sample, and then come back into the lab, take a look at the membrane under a microscope, and pick out the pollen grains. All right. Any others? Looks like we are just at the end here, but I'm glad to answer anything else. All right, I will end this then. Thank you, everybody, for coming.